may represent the sister group to all the angiosperm. Notice that the origin and the fossil itself. Notice that display both derived and primitive traits. Sinensis, for example, has anther and has seeds inside the closed carpels, but lacks petals and sepals. In 22, scientists completed a phylogenetic comparison of a sinensis with 175 living plants. Sinensis with 175 living plants was not included because its fossils are not as well preserved. The researchers concluded that among all known plant fossils, Archaeophractos sinensis is the most closely related to all living angiosperms. If Archaeophractos is indeed a proto-angiosperm, that would suggest that the ancestors of flowering plants were herbaceous rather than woody. Archaeophractos was discovered along with fish fossils and has bulbous structures that might be aquatic adaptations, implying that perhaps angiosperms originated as aquatic plants. The discoverers of Archaeophractos have suggested that fast-growing herbaceous plants could have returned to water and thereby thrived their escaping competition with other used plants and then later taking over the land. More recently, however, some scientists have disputed that interpretation. More derived lineages of angiosperms also become aquatic, and along the way the flowers became simpler, resembling the primitive flowers of Archaeophractos. Such debates, typical whenever a seemingly transitional fossil is discovered, may be resolved when more fossils and other types of evidence emerge. An Evo-Devo hypothesis of flower origins. Chapter 25 discussed how studying developmental genes informs hypotheses about animal evolution. The same holds true for flowering plants. Scientist botanist Michel Forge of the Natural History Museum in London has used the Evo-Devo approach, the synthesis of evolutionary and developmental biology, in hypothesizing how pollen-producing and ovule-producing structures were combined into a single flower. He proposes that the ancestor of angiosperms had separate pollen-producing and ovule-producing structures. Then, as a result of mutation, ovules developed in some microsporophytes, which evolved into carpels. Because Froelich asserts that the flower evolved mainly from the pollen-producing male reproductive structure of a gymnosperm ancestor, his view is known as the most male. Hypothesis supporting evidence includes comparisons of genes that give rise to flowers and cones. Flower developmental genes are usually related to pollen-producing gymnosperm genes. Also, certain mutations cause flowering plants to grow. Let's talk about human welfare. Depends greatly on seed plants. Throughout this unit, we have been highlighting the important ways to which humans depend on various groups of organisms. No group is more important to our survival than seed plants. In forests and on farms, seed plants are key sources of food, fuel, wood products, and medicine. Our reliance on them makes the preservation of plant diversity critical. Products from seed plants. Most of our food comes from angiosperms. Just 6 crops: wheat, rice, maize, potatoes, cassava, and sweet potatoes yield to 80% of all the calories consumed by humans. We also depend on angiosperm to food livestock. It takes 4.8 kilograms of grain to produce a kilogram of grain-fed beef. Modern crops are the products of relatively recent periods of genetic change resulting from artificial selection after humans began domesticating plants approximately 13,000 years ago. To appreciate the scale of the transformation, contrast the size of domesticated plants and their smaller wild relatives. As in the case of maize and Ticinti, at the genetic level, scientists can also glean information about domestication by comparing the genes of crops with those of wild relatives. With these dramatic changes, such as increased crop size and removal of the hard coating around Ticinti kernels, may have been initiated by as few as 5 gene mutations. How did wild plants change so dramatically in such relatively little time? For thousands of years, farmers have selected the seeds of plants with desirable traits, large fruits, for example, to plant for the next year's crops. But the humans may also be domesticating certain other plants unconsciously, as in the case of wild almonds. Almonds contain a bitter compound um, amygdalin that repels bears and other animals. Amygdalin, notice that no, okay. Amygdalin breaks down into cyanide, so eating a large number of wild almonds can be fatal, but mutations can reduce the level of amygdalin, making almonds sweet rather than bitter. White bears eat almonds from trees with such mutations. According to one hypothesis, humans may have absorbed bears eating the almonds and then eating the almonds themselves, passing some seeds in their feces. The seeds germinated around human settlements and grow into plants with sweeter, less dangerous almonds. In addition to basic crops, flowering plants provide non-staple foods. Two of the world's most popular beverages come from tea leaves and coffee beans, and you can think that the tropical cacao tree for cacao and chocolate. Spices are derived from various plant parts such as flowers, cloves, saffron, fruit and seeds, vanilla, black pepper, mustard, cumin leaves, pisal, mint, sage, and even bark, cinnamon. Many seed plants, both gymnosperms and angiosperms, are sources of wood, which is absent in all living seedless plant. Wood consists of an accumulation of tough-walled xylem cells. Wood is the primary source of fuel for much of the world, and wood pulp, typically derived from conifers such as fir and pines, is used to make paper worldwide. Wood also remains the most widely used construction material. For centuries, humans have also depended on seed plants for medicine. Many cultures have a long tradition of using herbal remedies, and scientific research has identified the relevant secondary compounds in many of these plants, leading to the synthesis of medicines. Willow leaves and bark, for instance, have been used since ancient times in pain reliever. Notice that this is the table lists some medicinal uses of secondary compounds of seed plants. Okay, back to the medicine also, or the prescription. Have been used since ancient times in pain relieving remedies, including prescriptions by the Greek physicians Hippocrates in the 18th century or years. Scientists traced the whale's medicinal property to the chemical silicon synthesized derivative acetylsalicylic acid is what we call aspirin. Although modern chemistry facilitates a laboratory synthesis, plants remain an important direct source of medicinal compounds in the United States. For example, about 25% of prescription drugs contain one or more active ingredients extracted or derived from plants, typically from seed plants. Other ingredients were first discovered in seed plants and then synthesized artificially. This lists some medicinal uses of secondary compounds of seed plants. Notice that compound atropine, and example of source belladonna plant, and the example notice also piabulditor in eye exams, the example of use. Another compound digitalin methanol or digitalin foxglove and heart medication. Menthol. Notice also the example of source eucalyptus tree, and example ingredients in cough medicines. Notice also morphine, opium poppy, pain reliever, and quinine. Notice also the example of source as tree. Notice also the quinine, Taxol tuberculari, and the curare tree, and vinplastin, periwinkle, and the leukemia drug Ethiopia curare, the example curare tree, muscle relaxant during surgery, and Taxol pacific U. Example ovarian cancer drug, ovarian, and pacific U. Okay, quinine. Notice the quinine cinchona tree. Notice also malaria preventative. Okay. Threats to plant diversity. Although plants may be a renewable resource, plant diversity is not. The exploding human population and its demand for space and resources are extinguishing plant species at an unprecedented rate. The problem is especially severe in the tropics, where more than half the human population lives and where population growth is fastest. 50 million acres of tropical rainforest, an area about the size of the state of Washington, are cleared each year. A rate that would completely eliminate Earth's tropical forests within 25 years. The most common cause of this destruction is slash and burn clearing of forests for agricultural use. As forests disappear, so do thousands of plant species. Once a species becomes extinct, it can never return. The loss of a plant species is often accompanied by the loss of insects and other rainforest animals. Researchers estimate that habitat destruction in rainforests and other ecosystems is claiming hundreds of species each year. This rate is faster than in any other period, even during the extensive Permian and Cretaceous extinctions. While the toll is greatest in the tropics, the threat is global. Many people have ethical concerns about contributing to the extinction of living forms, but there are also practical reasons to be concerned about the loss of plant diversity. So far, we have explored the potential uses of only a teeny fraction of the more than 290,000 known plant species. For example, almost all our food is based on the cultivation of only about 24 species of seed plants, and fewer than 5,000 plant species have been studied as potential sources of medicines. The tropical rainforest may be a medicine chest of healing plants that could be extinct before we even know they exist. If we begin to view rainforests and other ecosystems as living treasures that can regenerate only slowly, we may learn to harvest their products as sustainable rates. What else can we do to preserve plant diversity? Few questions are as important.